Okay. Hi, everyone. Thank you for looking at the video for the problem solving session 7 for the course Animal Physiology, which is an NPTEL course where we are so far into the week 7. And for the past 6 weeks, we have been discussing several topics on animal physiology. And today we will continue our discussion from the previous session. So if you remember in the previous week, we discussed about, so in the previous few weeks, we have been discussing about neuromuscular junction, how does nervous system function, what are the different types of nervous system that you have, how are they cross-linked, what is the skeletal muscle and everything together, right? So today we are going to see when the normal functioning doesn't take place, what are the type of problems that arise. So basically we're going to discuss about neurodegenerative disorders. And if you remember, we had discussed in week 7, this was part of your syllabus where you had 5 lectures and the lectures followed from one disease to the other. So it started with Alzheimer's disease, and then we moved on to Parkinson's disease. Then we had ALS, which is amyotrophic lateral sclerosis. We had spinal cord injury, and the last topic or the last class was about glial cells or glial cells. So I am going to talk to you about all these different disorders and diseases, um, like in the beginning. And then, like always, we have a set of questions that will help you understand these topics much better. And as and when you solve more questions pertaining to these disorders, the mechanisms of the disorders, the pathophysiology of the disorders, how can it be treated, what are the associated problems, these hopefully will become more clearer to you as you solve more questions. So let's try to look into what the first disorder is. So the first disorder, if you remember, in the lecture notes, we started with Alzheimer's, and it started with the story of Enoch Wallace, which basically was about a middle-aged person who started having memory loss. So memory loss, in our day-to-day life also, we keep saying about I have memory loss, this person has short-term memory loss, this person has long-term memory loss. So when we are talking about Alzheimer's, we are very serious because this disorder is the it's the actual process of dementia when you start forgetting things. And if you go into the brain structure about for a healthy person and an Alzheimer's person, and if you remember from the lecture videos, uh, we can clearly see a difference between the structure of the Alzheimer's patient's brain and healthy person brain, which you can see in this image as well. So you can see how the brain has started to shrink, and you have these different bodies here. So we'll get to this, what are these different bodies. So basically, if you have to define Alzheimer's, it's basically a neurodegenerative disorder, a progressive neurodegenerative or disorder, and it's mainly the cause for dementia. Dementia means memory loss, right? So what all gets affected? If you remember, it affects your memory, it affects your cognition, and it affects your behavior. So basically, it doesn't just affect your memory, it also starts affecting your identification. So you start having problem with your cognition, you start having problem with your behavior. So these are the different symptoms that you see here. So you have memory loss, you start having problems in communication, you have problems in difficulty in problem solving, planning, very confused state. There's how much changes in the behavior in terms of the mood and the personality. So all of these different changes are observed in Alzheimer's. So you can basically club them into two main category: memory loss and cognitive decline. And along with this, you see behavioral changes. Now, when I told you about these bodies here or these kind of formations here, these are basically the amyloid beta plaques. So these, when you see the formation of these plaques, these are the giveaway that the person is suffering from Alzheimer's. And that's when, if you go to a doctor with a memory loss as your symptom, then the first thing they ask you to do is scan for your brain and look for these bodies and start to see if you are suffering from dementia. So it's the telltale sign of Alzheimer's disease. And along with this, you have several other problems that is present in the Alzheimer's patients. Not only these plaques, but you have tau protein accumulation, you have neuronal loss. So the why we are connecting here with neural system, because along with the brain problem, you see neuronal damages in the diseases that we will be discussing today. We will see different kind of neurons are getting affected at different diseases. So it tells you that different neurons are leading to these kind of diseases. And when we are talking about Alzheimer's disease, we are talking about acetylcholine neurons. So those neurons get affected, and that's when you have Alzheimer's disease. So your neurotransmissions get affected, and that's how you have all these different difficulties in memories and your cognition, right? Right. So I will not talk about it much because I have a lot of questions. We can do that questions and it becomes easier for us. So the next disorder, which is relevant for today's discussion, is Parkinson's disease. Now, remember the previous one, Alzheimer's is a dementia disorder. So it has more to do with your cognitive changes, your memory changes, your behavioral changes. But Parkinson's mostly deals with these motor problems, right? So if you see a Parkinson's disease person or Parkinson's patient, you will see these kind of symptoms, which include tremor. So tremors are basically shaking of your hand or shaking of your body, right? So you will see tremors. You will also see closure of eyelids, constipation, sweating. These are all the symptoms that you can associate with Parkinson's. Tremors is basically the you can say one of the most visible and obvious sign of Parkinson's. And why it happens and what are the other effects? So basically, it diminishes your substantia nigra, which is your part of your brain. And again, like I said, because we are discussing about neurons here, so your neurons are also affected in Parkinson's. Which neurons? Dopaminergic neurons, right? So the dopamine neurons are the ones that get affected. So the dopamine producing neurons, which are part of the substantia nigra, they die off. So the neurons die, so the neurons get disrupted. And as a result, your dopamine transmission is not taking place between the neurons. And dopamine is very important for your muscle movement. So your muscle movements are, if you remember when we were talking about dopamine as a neurotransmitter, we were talking about them being regulatory in nature. So they are basically controlled, they're responsible for regulated, controlled, smooth muscle movement. Now, if there is no dopamine signaling taking place or dopamine transmission taking place, that very controlled movement is getting affected and you start having these shaky behavior. Your hand starts tremoring. So that's why tremor is basically the telltale sign of Parkinson's disease. Along with this, just like in your Alzheimer, where you had those plaques, in this you will have something called Lewy bodies. And with all of these motor neurons, motor symptoms, you should also remember that even in Parkinson's, you will have non-motor symptoms. So in Alzheimer's, you only had non-motor symptoms. But when you come to Parkinson's, you have first of all these motor neurons or motor symptoms. The next ones are non-motor. When it reaches advanced stages, you have cognitive problem, you have mood disorders, you have sleep disturbances. All of these are the non-motor, which something which does not involve your movement, but those come at the extreme stages, right? Now, because unlike Alzheimer, this is more of a dopaminergic neuron. You are targeting, you can directly target those dopamine. So we have come up with a treatment. And if you remember, the treatment was L-DOPA treatment. So which is also called levodopa treatment. So basically what you are doing is, because your dopamine is getting affected, you are like the doctors are giving dopamine precursor. So the dopamine precursor, what it does is it crosses the blood-brain barrier, and then the transmission which was not taking place is then taking place. You have the dopamine in place, and then it starts doing the regular activities. So you can treat this using this L-DOPA treatment, but you will realize that blood-brain barrier is the most important part for any of these treatment, right? Now, what is blood-brain barrier? Your brain is not very easily accessible by each of the components that enters your body, for obvious reason. You want your brain to be protected. You don't want your brain to get exposed to every other thing that comes into the body. So your body has made it very it's ensured that your brain is sufficiently protected, and there is a blood brain barrier which needs to be crossed for anything to reach to brain, right? So we call this as blood brain barrier, and it is very selective, semi-permeable boundary, and it separates any circulating blood from this brain. So it doesn't allow the contact of brain with these molecules. Now, like I said, it's important for protection, it is important for nutrient transport. So it's very selective in nature. But what happens in these disorders are these very blood-brain barriers get disrupted. And because it gets disrupted, you can imagine, because your barrier is getting disrupted, you have a lot of these neurodegenerative disorders, because something which is encasing the brain, something which is protecting the brain, is now disrupted and it's affecting the brain. So blood-brain barrier disruption is something which is very important with respect to neurodegenerative disorders, right? Okay, so the next one that you had discussed or we had looked into in the lecture notes, what's ALN? ALS, which is also called amyotrophic lateral sclerosis. Now, this is again a neurodegenerative disorder, and this affects the motor neurons. So this is primarily motor neurons in both brain as well as spinal cord. So till now we were talking about brain, till now we were talking about movements. Now we have a disorder which is pertaining to both brain and spinal cord. Why is all of this important for you to remember? You need to remember it that we are looking from a neural system perspective. So we're going to cover all the disorders that are pertaining and important for the neural system. So we have done Alzheimer's, we have done Parkinson's. Now we will look into ALS, which is basically the degeneration of the motor neurons of both the brain and the spinal cord. And what it leads to is loss of your muscle control. So over time, your muscle gets atrophied, your muscle problem starts showing up, right? So as you can see here, this is a normal nerve cell. So you have nerve cell coming and attaching to muscle cells, and this is your proper conduction. But when your neuron starts getting affected, this muscle is not being in use anymore. It gets atrophied. It is a wasted muscle. So this is ultimately affecting your musculature as well, right? So also your in your ALS, both the motor neurons and towards the later stage, your sensory neurons also get dysfunctionized, right? And you have treatment for both of these, but how far we have gone, we will see in the questions as we solve. But one of the most important mutation that is responsible for this, if you remember, was SOD mutation. Now, what is SOD? SOD is the name for superoxide dismutase. Okay. Now, mutation in general means that something has gone astray from its normal activity. So let's first understand what is dismutase normal activity. So what is dismutase? Or basically superoxide dismutase? It is basically an enzyme, as the name suggests from the word ACE, and this enzyme is responsible for breakdown of superoxide radical. Okay, and it breaks down the superoxide into oxygen and hydrogen peroxide, which is further broken down by catalase into water and oxygen. So you're basically, why are you cleaving this superoxide? Because these are something called ROS. These are called reactive oxygen species. What you need to remember is these are highly reactive. So in your cells, in your body, if you have these superoxides or high free radicals, these will basically react and cause a lot of cellular instability. So you don't want that. That's why the cell, the body, tries to remove all these superoxides, tries to remove all of these ROS. So all these reactive oxygen species. Every species is basically all the groups which have reactive oxygen. So anything which has reactive oxygen is something that we don't want in the cell. So you want them to go away, right? Now, this is for the superoxide class. You have this enzyme called superoxide dismutase. It removes this superoxide. But if you have a problematic superoxide dismutase, if your enzyme itself is problematic, your superoxides don't get removed. And because your superoxide don't get removed, they do every harm that is possible for your cell. So you see, that's why your cell starts getting affected, because now your superoxides are not getting removed anymore. So they start getting So these superoxides start getting aggregated, they become toxic in nature, and in neurons they start killing the neurons, they start affecting the neurons, they basically impair the neurons. So once the neurons are affected, you have these muscles which are no more interacting with the neurons, they start getting wasted, so your muscles start getting atrophied. You will see there is a curvature, your muscle starts getting loosened up, it becomes atrophied. So all of these are because of ALS. So we have done the third one also. And the last one, which was discussed, was spinal cord injury. And why is it like, why did we discuss it separately? Because spinal cord, if you know, in any accident, spinal cord is the most easy to get harm because you're falling on it, you have a direct exposure. So spinal cord injury is also equally important for us to know what are the pathophysiologies. So when you call anything related to spinal cord injury, you're basically talking about, because remember all the neurons are here, everything is being controlled by brain and spinal cord. So unlike brain, which is highly encased in here, you have spinal cord which is right here. So any injury to spinal cord affects your motor, affects your sensory, affects your voluntary muscle control, everything, right? So these are mostly caused by trauma, any kind of accident, any kind of fall, any kind of violence, anything, trauma. So trauma can also cause spinal cord injury. However, you don't always rely, or you don't need always the violence or trauma. There can be non-trauma also, and that's because of cancer. So you have these tumors, you can have infections, or you can also have these degenerative disorder that we just discussed in the previous part also, right? So what basically happens in a spinal cord injury is, you have first, the initial damage to spinal cord basically leads to the compression of spinal cord, because of course it's getting affected. You have laceration, so you have some kind of injury marks. You have neuron separation, because neurons are innervating your spinal cord, and if a spinal cord is getting affected, your neurons are getting severed. Severed is basically detached or removed from that part. But that's just the primary. What follows from all of these problems are way problematic. Your secondary injury is what begins the actual problem. You have inflammation, so your body starts to basically produce immune cells, and the immune cells go there and cause a lot of inflammation. You have your disruption of blood brain barrier, because I told you that your blood brain barrier starts getting affected as soon as your degenerative disorders kick in. You have free radical generation, you have something called glial scar, and we'll see what that is. You have cell death, you have all these problems. So what are these terms here? If they are on the upside, they're basically showing what should not be the case. But right now we are discussing about spinal cord injury. So we are discussing that what all we are seeing in an injury, and how does this manifest? How do you realize this is present in a patient? You see them paralyzed. So their spinal cord gets paralyzed. They so basically you will see them either not moving their left part, right part, lower part, upper part, not able to get up. So paralysis is the outcome, or paralysis is the clinical consequence of your spinal cord injury, right? And there was a term called glial scar, which is pertaining to glial cells. Now, what are glial cells and why are we discussing here? So your glial cells, if you remember all the cells that we discussed in the previous classes, glial cells are basically cells other than the neuronal cells in your nervous system. So you have all of these different kind of cells. If you look in your PNS, you have satellite cells and squamous cells. If you look in your CNS, you have microglial cells, you have ependymal cells, oligodendrocytes, and astrocytes. All of these do their own respective role. For example, your astrocytes here, they support neuron, they maintain the blood-brain barrier, they regulate neurotransmitters, right? The oligodendrocytes, which are basically responsible for myelination of CNS neurons. They remember we spoke about myelination and the importance. So these cells are what are responsible for the myelination of the neurons. So they are myelinating the CNS neurons. Similarly, if you have oligodendrocyte in CNS, you have squamous cells in PNS which myelinate these neurons. Okay. Then you have this microglia. These microglia basically the immune cells of CNS, the cells that I just told you, when it is responsible for the inflammation in response to an injury, right? And the last one, these ependymal cells, these are responsible for again maintaining the hemostasis. So they produce something called cerebrospinal fluid. So basically, it's part of the anatomy and it is responsible for giving structural stability. So they and why are we discussing here suddenly? Because they are all having a role in each of the neurodegenerative disorder that we study. Because the body wants to be in such a condition that they can reverse the problem that is faced. So let's say you have Parkinson's, or let's say you have dementia, Alzheimer's. So the body would like to use these, like you can say cell warriors, or you can say these ammunitions or tools to reverse the problem. So these are the cells that take part in the initial stabilization of the issues. But when you reach at a stage that even these cells are not able to function anymore, that's when your problems start cropping up and your symptoms start becoming more obvious and you degenerate over time. And that's when it is called the neurodegenerative disorder. So that's why we need to understand that even glial cells also get affected, even though they are not the neuronal part of the nervous system. They are not the neurons that is getting affected, but because they have additional role, you should also know about them. So that's what you had looked into the week 7. And I rushed through it because I want to discuss all these questions that we have, and hopefully as soon as we go ahead by answering the questions, we can answer this better, right? Okay, so let's begin the question so that we don't start getting too bored with the discussions already. Okay, so let's try to understand first the non-disease part of this week's course. So let's start with answering different kind of questions. So first is electrical conductivity of neuronal cells would dash in the absence of myelination. This is just for your how do you say, refresher of whatever we did so far in the previous session. So basically this question in other words is asking you, why do we need myelination? So if you don't have myelination, what happens to your neuronal conductance? And this, if you remember why we need myelination, this is basically in relation to that. So if you have neurons, you have myelin sheet. So that's basically giving them the jump for the transmission to take place. And because they are jumping, it's faster. So if you don't have myelination, it basically does the opposite. Would you like to attempt the answer, or else we can move ahead because we have a lot of questions? Okay, as a rule, let's just wait for 1 minute in each question so that we reach, because I want to discuss the diseases which are more which I want you to have more practice of so that you understand the diseases much better, right? Okay, so let me move ahead with these questions because these are just for your refresher. So like I said, as you don't have myelin sheath, so it has a longer route to travel, so the speed of the conductivity will be decreasing. Okay. Now, which of this property of the neurons, which of these is the property of neurons that carry pain signal? Remember, we are talking about neurons which carry pain signals, and nociception. So you have pain signals. Do you want the information to go quickly, or do you want it to be long and sustaining? You can think in this way. So the question here is saying that what are the property of the neuron? So you have your options are they have huge level of myelination, they have no cell bodies, and they are unmyelinated. So you need to think in terms of how do you want the transmission to be? What would be the ideal scenario of a pain transmission? You have a pain, if you have quick pains, quick pulses of pain, do you want that? Is that the optimal condition, or do you want it to be slow and steady? So while answering this question, you can think from that perspective. Okay. So let me give you the answer so that we move quickly. So these are unmyelinated, like I mentioned. Now, why do you need these unmyelinated? Because you want slow signal conduction. And why do you suddenly want slow signal? So far, we were saying fast signal, we need it fast, we need to reach it fast. Because here we're talking about pain. We want that pain to last longer, diffuse across, so that the signal is received by each and every cell, and this the pain is, we are able to address the issue of the pain. If it is fast and quick, we might not even register, and we might not even react to it. So we want it to be long lasting, we want it to diffuse, we want the cells to come into play, we want everything to take care of the source of pain. So you want it to be long lasting, that's why we want it. The neurons would rather be unmyelinated. Okay, think of it like that. Okay, now the next thing was something that we discussed, that you discussed or would have seen in the lecture notes, basal ganglia. So out of these options, which one do you think is the primary role of basal ganglia? And why should we know these names? Please remember, these names are very important because the subject is animal physiology, and you need to be very careful about which part does what, right? So especially in brain, where you have so many complicated names, these are the few common names and easier names that you need to remember. For example, basal ganglia. So which of these is a function of basal ganglia? You should try and remember that. Right. So let me give you the answer so that we can move to the next question. So basal ganglia are basically the one which are important for regulating and coordinating movements by modulating cortical motor output. So complicated what basically just means that they are regulating your movement by controlling the motor commands, how to move your, to the commands that go for the motor neurons. Why did we read this? Why is it important? Because it tells you that all the diseases that affects the motor neurons or your motor capabilities in these get affected in the disorders are basically targeting the basal ganglia. That's why you should remember which part has what role. It goes both way. Either you remember, okay, Alzheimer's, let's say, for example, or let's say Parkinson's is affecting basal ganglia. It's either you remember it this way, that okay, if as Parkinson's is affecting the basal ganglia, and we know Parkinson's have problems in motor neurons, that means basal ganglia is part of motor neurons, basal ganglia is controlling it. Or you remember the other way, you remember that okay, basal ganglia is taking part in controlling motor neurons, and our Parkinson's are affecting basal ganglia, so they are basically affecting the motor neurons. So that's why you need to know all these components so that you can, like mix and remember them. Okay. Right. Okay. Which are the following pathways is involved in transmitting movement related signals from the brain to the muscles? Again, just like the previous question, this is important for us to understand the pathophysiology of your disease. So if you have any disorder which affects the movement signals, which motor pathways are they basically targeting? And this we have done in the previous class also. When we looked into the classification of ascending and descending, we told which one is pertaining to sensory and which one is pertaining to motor, if you remember, and which would be getting affected. This one is very easy. If the sensory and the motor word would have been removed, it would still be easy, because when we had discussed in the previous class, we basically saw a map of how this works out, right? But here in this question, it's basically giving away the answer right at the word level. right? So like I said, it's about the descending motor pathways because we are talking about movement. So anything which is movement is motor pathways. Okay, now very important. Dash may be released as a transmitter in neuronal preparations or as a mediator of an inflammatory response by mast cells. Okay, so basically it tells if you have neuronal preparation, which could be a transmitter. Now, all of these are the ones, but it is also saying that it is taking part in inflammation by mast cells. Now, if you remember your immunology classes and you remember these cells, it will become very easy for you to guess the answer just by the two words there. One is mast cell and one is inflammation. Which one do you think takes part in these responses? Let me wait for you to register and try to answer these questions. Do you have a guess? You can take a guess here. So we have acetylcholine, we have glutamate, you have dopamine, and then you have histamine. So which of these words you generally associate with allergy or inflammation in general? Any idea? Okay, no worries. I will show you the answer. So it's histamine. So whenever you see mast cells, just remember immunology classes, mast cells or inflammations are basically for histamine. So histamine has a role in those kind of responses. Okay, now these we have discussed in the previous class also. We have looked into hippocampus and what kind of neurons they have. So basically the following statements here say hippocampus contain pyramidal neuron, interneuron, astrocytes, and oligodendrocytes. And the options then say which of these statements are correct. So which of these neurons are basically present in the hippocampus? Any idea which of these are present in the hippocampus? Okay. So basically here all of these are present in the hippocampus. We have seen it has pyramidal neurons and we discussed pyramidal neuron extensively in the last class. We also discussed interneurons. We looked into astrocyte and oligodendrocyte right a few minutes back here in this session and the previous session as well. So let me also ask you from this only, which of these can myelinate multiple neurons? There are two words here. First of all, we want cells which myelinate neurons, and second, they myelinate multiple neurons. So that I will wait for you to see the question. And if you want, if you feel like that you know the answer, please type in the chat box or think of an answer, and then I'll give you the answer for this. And these are very easy questions to remember. These are objective questions that help you understand the topic much better. Okay, let me give you the answer. So oligodendrocyte. Okay. Now let me just connect these with the things that I have told you in the beginning. So remember I showed you the cells. Let me show you the cells again. Just bear with me. Yeah. All these cells, I told you what all they do, right? Out of those, I told you this cell and this cell myelinate the axons. Axons which are part of CNS, axons that are part of PNS. So they myelinate. Basically, they produce the myelin sheath on the axon. But what is very interesting, and that's how you can use it to remember these different cells, is the way they myelinate the axons, right? So if you, the question here was, which one of these cells myelinate multiple axon? So the answer, as I told you, was oligodendrocyte, and you can see oligodendrocyte is part of CNS. So what is happening in central nervous system is when they have these neurons to, so this was discussed in your lecture notes, if you remember, towards the end, professor had discussed. So you have these multiple neurons, and that means multiple axons, and all of them can simultaneously get myelinated by the oligodendrocyte. So a key for you to remember this, if it is difficult for you to remember, so oligo generally means multiples together. So you can remember it like this: oligonucleotides, oligodendrocytes, so multiple together, oligos multiple, and then you myelinate multiple axons, right? So in CNS, oligodendrocytes myelinate multiple axon. The opposite is done by Schwann cells. Your Schwann cells, which are in PNS, they are basically at a given time they myelinate only a single axon, right? So that you have, there are two cells that myelinate the axons, and there are very clear cut mechanism for both of them to undergo this process. And if you remember, the professor had told how they decide which neuron will be getting myelinated is very random, and there's no clear cut understanding for this, but the procedure, the mechanism is very different. One does myelination for multiple axons together, one only does for a single axon at a time. So that's a way that you can remember. And what about the other cells that were part of the question? So we had astrocytes and microglia, and we had discussed astrocytes are basically for your structural support that I told you. And microglia, as we have discussed, that it is part of immune response. It does not participate in myelination. So when we are talking about myelination, we are only talking about oligodendrocyte and astrocytes. Okay. Next one, which of the following correctly differentiates Schwann cells from oligodendrocytes? Now that we have already discussed this multiple times, let me read out the option for you, and I am sure as you read the options, it becomes easier for you to understand. So the first one says Schwann cells myelinate multiple axons. Oligodendrocytes myelinate a single axon. Schwann cells are in CNS. Oligodendrocytes are in the PNS. Schwann cells myelinate a single axon segment. Oligodendrocytes myelinate multiple axons segment. Schwann cells are electrically active, oligodendrocytes are not. So if you remember from the discussion that we have just had, you can answer this easily. Now, because we have just discussed this, I will wait for 30 seconds for you to first read and look at all the options and get to your answer. Right? Okay, so let me give you the answer. Right, I am sure if you would have read, you would have got it very easily. So if you have Schwann cells, the first one says Schwann cells, multiple axons, it's not correct. We just discussed the previous question. It's oligodendrocytes, which is myelinating multiple axons. So it's basically the opposite. Schwann cells are present in CNS, which is also incorrect because we just discussed oligodendrocytes are present in CNS. So this is also opposite. This one is therefore correct. Schwann cells myelinate a single axon, oligodendrocyte myelinate multiple axons. And Schwann cells are not electrically active, so this is basically incorrect. These Schwann cells are basically not actively not participating in action generation, activation or generation, action potential generation. I'm sorry. So this option is also incorrect, right? Okay. Next, what is the primary role of myelin sheath around axons? So this we have discussed so many times. All these are basically just a way of making this topic simpler for you. So I will wait for 30 seconds because this is an easy question we know already. So let me read out the options for you. So the first one says they are important because they synthesize neurotransmitter. They are important because they store calcium ions for synaptic transmission. They are important because they increase the velocity of action potential conduction, or they are important because they provide nutrients to synaptic cleft. And we know the answer should be increasing the velocity of action potential conduction, because they are basically allowing the action potential conduction to jump across the nodes of Ranvier, and because they are jumping, their speed is greatly increasing. Okay, next. During myelination, which cells are responsible for wrapping their processes around multiple exons? So this is another question based on the same type of question that we have just answered. This shouldn't take you much time to answer because we have just discussed it. Right? So we know it's already oligodendrocytes. Okay. Now, which of these cells are electrically inactive, which basically is saying that these cells are not responsible for generating action potentials? Very, very simple, because everything else so far we have discussed, and the first option here we have already looked into it, right? So glial cells, if you didn't remember by glia, if you remember the constituents of glial cells, which are basically astrocytes, which are oligodendrocytes, Schwann cells, everything that we have discussed so far, we have just looked into the option which said Schwann cells are electrically inactive. So basically glial cells are inactive, and this can also be a thought like this, that skeletal muscles are generating action potential, neurons are generating action potentials, and cardiomyocytes, which are basically cardiac muscle cells, are also generating action potential. So anything which is generating an action potential is electrically active. Glial cells are not, because they are mostly concerned with structural support, metabolic support, myelination, and immune defense. So these are the other accessory functions which are there in the nervous system other than the neuronal cells. Okay. Okay. So like I said, if that's not the correct option, which is the correct option for glial cells? Are they conducting action potentials? Are they providing structural and metabolic support? Are they electrically excitable, or are they synthesizing neurotransmitter exclusively? And we know that, just how I have just explained, they are providing structural and metabolic support to neurons. The other three options are basically about the other neurons and cells. These are not concerned with glial cells, right? In addition to these two, glial cells have many more roles, but none which pertains to neurotransmitters or action potentials. Okay. Okay, so that brings us to an end of the part which discussed more about glial cells and other cells which are part of your neural system. Let's move our gears and start discussing about diseases. And my only attempt is that you understand or get a better understanding of all these diseases that we will discuss today. Right? So the first question here is, dash affects memory because of degeneration of mostly this. So what is the key term here? Memory. So if it is about memory, which disease, and if that is memory and that disease is known to you, then which neurons are getting affected. So like I said, look at the key term. The key term here is memory. And we have discussed the disorders which are related to memory. So think of it like this and then answer the question. Okay, let me give you the answer. Like I said, it's memory. It's Alzheimer's disease. And because it's memory, the neurons are getting affected in the hippocampal region. Okay, so discussing more about Alzheimer's disease. Alzheimer's disease primarily affects which brain region responsible for memory formation. So we know Alzheimer's is for memory and it's affecting brain, but which of these region mostly primarily? And which neurotransmitter is affected for Alzheimer's disease? I've discussed both of these in today's session. So if you can remember, I will wait for certain seconds. Okay, so let me give the answer. Hippocampus for the region of brain, because we have just discussed in the previous part also, because we have more. So Alzheimer's concerns memory, and that's part of hippocampus and the cortex. And the neurotransmitter, which was part of Alzheimer's, is acetylcholine, right? So that transmission of your acetylcholine gets affected in Alzheimer's. We will discuss the other neurotransmitter when we come to the next disorder. Okay. Now, I've already discussed about telltale signs of Alzheimer's. It is also called hallmark pathological feature. Hallmark means the definite sign of any disease or anything for that matter. So here it says, which of the following is a hallmark pathological feature of Alzheimer's disease? So if you see that in your diagnosis, you for sure know that the patient is suffering from Alzheimer's. So which of these is those feature? Remember that we have had this discussion. I told you which one is the hallmark feature of your Alzheimer's. And along with this, the other options also with per part or some of it I had discussed in the discussions today. Yeah, if you remember, amyloid beta plaques and the neurofibrillary tangles. So basically, all these plaques of amyloid beta, as the name suggests, these are plaques. So these are basically extracellular deposits. These are for the amyloid beta protein fragments. So the beta protein gets broken down and the fragments get deposited outside the cells and they accumulate between the neurons. And that's why the neuronal transmission gets affected. And then the other one is neurofibrillary tangles. So as you remember, I told about tau protein. So these tangles, as the name suggests, these are tangles, right? So these things are getting interspersed with each other. So these are intracellular deposit of tau protein, which are hyperphosphorylated. So there are a lot of phosphorylated groups on the tau protein. So these hyperphosphorylated tau proteins are accumulated intracellular, and because like the ones which are these ones are extracellular, so they are disrupting the transmission. These are intracellular, so they're disrupting the neuronal structure, the neuronal cytoskeleton, or the neuronal cell structure. So the neurons are therefore getting impacted both from outside and inside, and that's why they are dying off or they are degenerating over time. And that's the reason for your neuronal death in Alzheimer's disease, right? So amyloid beta plaques and neurofibrillary tangles. The other thing, Lewy bodies, if you remember, we discussed this in Parkinson's. Demyelination is basically something which is related to a disease, a problem called multiple sclerosis. And dopaminergic neurons, dopamine is again for Parkinson, because Alzheimer's are for acetylcholine. Or acetylcholines are the ones which are getting affected in Alzheimer's. Okay. Okay. Now, if we are talking about Alzheimer's dementia, which of these problems do you see associated with Alzheimer's? So, do you see impaired motor coordination? Do you see progressive memory loss and cognitive decline? Do you see increased procedural memory, or do you see enhanced sensory perception? Two out of these are very easy for us to rule out that this will not happen in dementia. But the other two, which one of these is present, or which one of these uh, symbolizes Alzheimer's dementia, as the name dementia also suggests? Okay, let me tell you the answer, which is progressive memory loss and cognitive decline. So as we know, Alzheimer's is disease, dementia. Dementia, the term itself says memory loss, right? So your Alzheimer's dementia is basically when you have progressive memory loss. It's basically your the how do I say, the entire process of you acquiring short-term memory and associating that for a long-term memory gets affected. Your short-term memory, which is the first step of acquiring memory, is starting to getting affected. So you have, because that is getting affected, you don't have long-term memory. So you have, there's a progressive memory loss, and you have cognitive decline. What cognition are we talking about? We're talking about reasoning, we're talking about judgment, we're talking about thinking, we're talking about language. So if you see Alzheimer's patient, it starts with memory loss, and over the time, that person has problem in thinking, remembering, reasoning, anything which is associated to with anything with memory. So all of that get affected in Alzheimer's. Now, the other options, which is increased motor coordination. If you remember, Parkinson's is the disease which is primarily associated with motor problems because of the tremors or the shaky hands that we have been speaking about. So motor coordination is a problem for Parkinson's. And enhanced sensory perception and increased procedural memory are not part of dementia. Dementia is the opposite. Dementia has loss of memory, and you don't really have any increased sensory perception. Okay. Right. Now, next. So we are talking about the early stages of Alzheimer's, which is the first thing that happens in Alzheimer's disease. Is it immediate loss of motor function, or is it complete loss of ability to speak, or it is hyperactivity of the hippocampus, or the subtle memory loss and difficulty recalling recent events? This is, so this, you can imagine this as a person who has just started to get dementia, just started to experience the symptoms of Alzheimer's? What is the first set of symptoms that that person will experience? Yes. You can use the chat box to type in your answers or any query that you have, because I would be in the other screen. So you can please use the chat box to type, or else you can, if you want to, you can unmute and tell what you want to say.
Ma'am,
Yes.
the chat box is not working, ma'am.
Sorry,
Chat box not working, ma'am.
chat box is not working. I'm not sure why, because okay, let me see.
You are no longer a participant.
Can you try joining in again?
Ah, yeah, ma'am.
Sure. Thank you. I think it should be working because it's working for me. Okay, while that's happening, let me give you the answer here. Right. So the first stages of Alzheimer's disease is subtle memory loss and difficulty recalling recent events. And why, how it becomes easier for you to answer this in this course? Because if you remember, the professor started discussing the entire Alzheimer's with a book, and it started by talking about how there was a patient in a middle-aged man who had difficulty recalling stuff. Suddenly he had problems remembering stuff, and that's the first sign of dementia. That's the first sign of memory loss. You suddenly stop remembering the things that you very much knew. You knew something, you have difficulty. This is also, I don't know, but the movie, there's a movie also, I think it's called something Alice or something, where basically that she's a professor, I guess, or a psychiatrist, and she goes up on the stage and she has to deliver a talk, and she's on the stage and she wants to talk, but she can't remember words. That's very unusual for a professor, but that's exactly what happens in your memory loss. You suddenly have confusion, you can't remember words that you are otherwise using in daily day-to-day activities, and you suddenly seem to have lost the information. So that's the beginning of dementia, is the beginning of your Alzheimer's, right? So your subtle memory loss, you have difficulty in recalling your recent events. Those are the problems with your Alzheimer's disease, right? And the other ones here, which has complete loss of ability to speak, comes way later. It's at the end stage when everything starts getting degenerated. One thing leads to the other, and you're towards the end, your motor function also start to get problematic. You have loss of ability. But your initial stages and the most pronounced effect of Alzheimer's disease is your memory loss, right? There is no hyperactivity of hippocampus because your hippocampus is getting affected. So your hippocampal activity is rather decreasing and not increasing. Let's go to the next question to see. Um, yeah. So we have discussed about neuronal degeneration. What is the mechanism? So you can remember these diseases in certain topics or certain subheadings. First, what is the problem? Like, what is the symptoms of the problem? Second, why are those symptoms happening? And third, how do you treat them? So it's easier for you to remember in these subcategories so that you don't mess up or you forget stuff. So if you remember, if you classify all your diseases in these three categories, you are bound to remember all the relevant information about disease. So you will take an effort to see if there is treatment there. You will take an effort to see, we know there is motor problem, we know there is memory loss problem, but why? So the why question comes in the mechanism part, which is here. And the first one is basically understanding, how do you know it's an dementia patient, or how do you know it's a Parkinson's patient, or ALS patients. So all of these, so if you classify your diseases in these three parts, then I think it becomes very easy for you to remember any of these diseases. And these diseases are not, they are also very relevant because we see these names almost every day in some or the other way. And it is very important for us to understand these, and early prognosis is very much helpful. Right. So the question here says that if you have Alzheimer's, what is the proposed mechanism? Any idea? In case your chat box is still not functioning, you can unmute and tell me the answer. If you want to attempt the answer, if you want to wait and see, it's also fine. Okay, let's look into the answer so that we can have discussion when we are discussing the answer. So the mechanism of Alzheimer's, if you remember, we discussed about amyloid beta interfering. What beta interfering we are talking about? So you have the word is, the term that we discussed was amyloid beta plaque. Plaque is, so what is plaque? I didn't say what is a plaque. A plaque is basically something like a deposition. You would see in people who have high blood pressure or hypertension, or uh, how you say it, thyroid, or something which is to do with fat deposition. The fat deposition is called a plaque or a plaque. You generally hear. And where the plaques are, the plaques are in the arteries, in the blood vessels. Like there's a layer coated. So the coating, the layer is called plaque, right? So in terms of your neural disorders, amyloid beta plaques are basically these deposition, as you remember from the previous question, they're extracellular. So you have these two neurons, let's say, and in between you have a plaque of a deposition of these amyloid beta proteins. And now because they are in the between of two neurons, the transmission is getting affected. So your neurons cell to cell communication is getting affected, your transmission is getting affected. And this is leading to oxidative stress because there is no response. So you have more of free radicals, which suppose that is not getting quenched. You have inflammation, you have so many problems associated that is leading to all the manifestations of the neural disorders that we see, right? So the primary reason is that there is impaired cell to cell connection, impaired neurotransmissions because of the plaque in between. This was extracellular. We also discussed intracellular, where you have these tau protein accumulation. They are inside the cell. So from the outside, they are not interacting, not able to interact because it's a plaque formation. Inside, you have these tau proteins which are getting accumulated, and it is affecting the cytoskeleton of the cell. So the cell structure is getting affected and the neurons are starting to degenerate, right? So remember it like that. Now, the other topics, the other options that are here, which are NMDA receptor, excessive receptor inhibition. Well, that's not the case. There is overactivation, you don't have neurogenesis, and you don't have calcium buffering. So for Alzheimer's, the most pertinent is amyloid beta interfering, and you need to remember it this way. Okay. Okay. So we have discussed about Alzheimer's, and I hope all these have tried to show you how we basically identify Alzheimer's and what are the associated problems with it. Now let's move on to next set of diseases. This one is talking about DASH affects movement because of degeneration of mostly. Like in Alzheimer's, in the similar kind of this is the question from your previous year's assignment. So the key here is basically the word movement. So the word movement should tell you which disease we are talking about here. Like in the previous one, similar kind of question you had. So let me try to show you the question that affects memory because of degeneration of mostly something. So memory was something that gave us that this is Alzheimer's. So here you have movement. So you need to tell me which one is the correct answer here. And once you have identified which disease it is, which neurons are getting impacted. We did this for Alzheimer's. Now do this for Parkinson's as well. B. Yeah. Okay. Okay. Any more answers? Anyone else? Okay. Let me show you. This is C basically. So like we have been discussing so far, just a key for you since you missed the beginning of the class. Uh, Parkinson's disease is basically associated with tremors. So it's basically about the hand shaking. So if you see a person with shaky hands who's, having motor neuron problem, that is Parkinson's disease. Alzheimer's is dementia, is memory. So whenever you have this movement, it's Parkinson's. And because we are talking about movement, we are talking about motor cortex neurons. So motor is the key term for Parkinson's. Okay, movement basically. Okay, let's try to see more associated terminology with Parkinson's. So now hippocampus is getting affected for Alzheimer's. We know that. Similarly, what is getting affected in Parkinson's disease? Mainly, and what are the other reasons which are getting affected in Parkinson's? So basically in this question, you are supposed to give me the most important region of the brain that gets impacted. And in this, you are supposed to give me what is not getting affected by Parkinson. You're free to take a guess, whatever you feel like is the answer. There is no judgment here. We are here to discuss. So I can discuss more easily with you if you uh, want to answer. If you, so basically, like I was mentioning, it is about Parkinson's disease. And yeah, I have got the response for one, I guess. So you can try to give me for the second one also. All right, let me give you the answer so that we discuss more. So remember, in Parkinson's disease, actually, when we were looking into the slide, I showed you that the key brain region that is getting affected is substantia nigra. So what you need to do on your behalf is you should remember these terms very, very clearly. It's Parkinson's, then it's substantia nigra. If it's Alzheimer's, it's hippocampus. Okay. And these are the main region. Along with substantia nigra, you have the basal ganglia and dopaminergic pathways, which is affecting in Parkinson. Because in Parkinson, the relevant neurotransmitter is dopamine. In Alzheimer's, the relevant neurotransmitter is acetylcholine. This is also how you can differentiate, right? What you don't get affected is cerebellum. Because cerebellum is not something that gets affected in your Parkinson's. In your Parkinson's, because it's about motor neurons, your basal ganglia, which is controlling the motor neurons, those get affected. Dopamines get affected, dopaminergic pathways get affected, and most importantly, substantia nigra gets gets affected. Okay, now I already told you what kind of neurons are getting affected. So this question deals with that. So in your Parkinson's disease, which type of neurons is mostly getting degenerated? This should be easy because we just discussed this. Which neurotransmitter is part of Parkinson's and which neurotransmitter is part of your Alzheimer's? We have already differentiated. And okay, wait, sorry. Yes, like I just mentioned, it's dopaminergic. So Parkinson's disease, dopamines are the neurotransmitter which are responsible. Great. Okay. Now, so we know in dopaminergic neurons, so for the dopaminergic neuron, there is death in Parkinson's disease. So if your dopaminergic neurons are getting death or dying, in Parkinson's disease, what is the manifestation or what is the result of this death? And that's how you connect your problems. So how do you see the Parkinson's disease, or how do you realize this in a patient out of these options that are given? So the options are: there is an increased voluntary muscle movement, there is an imbalance between excitatory and inhibitory motor control, there is enhanced cognitive abilities, and loss of sensory perception. Any answer? A. Okay. Any more answers? Okay. Let me give you the answer so that we are on time today. Right. Okay. So the first one says, or the answer here is imbalance between excitatory and inhibitory motor control. Okay. Let's see the first one, like someone mentioned. It says increased voluntary muscle movement. In Parkinson's, when I said tremors or when I said shaking, shaking is involuntary. You don't have a control. So voluntary means control. Voluntary is I get to choose, I get to control, I get to regulate. I have that constant decision. I can voluntarily choose to touch something. I can voluntarily choose to not do something. So that's upon my control. So involuntary is when you don't have a control. And in Parkinson's, if you have seen Parkinson's patient, they are basically, let's say they're trying to scoop up something from a bowl and take it into their plate. The motion will always be shaky and they will be spilling all over because they don't have a control over your hands. So the hands don't really follow the normal movements and there's an extreme shaking, right? So you don't have a control. So that's not voluntary. So that's why the first option is incorrect. And because we are talking about dopamine, and if you follow my previous session, you would have seen that we had discussed about all these different type of neurotransmitters. We discussed about acetylcholine, we discussed about serotonin, we discussed about dopamine, and we have seen there that dopamine is basically for regulation. And because in Parkinson's, your dopaminergic neurons are getting dying or dead, so your regulatory effect is getting impacted, and that's why you have this imbalance between excitatory and inhibitory motor control. So that's why there is no uh, connection or there is no balance between them, right? That's why this shaking is happening. Otherwise, if you remember, excitatory, inhibitory are basically, excitatory means to do something, inhibitory is to stop. So you have a control over it. You don't have a control of that in Parkinson's. And the other two are not the result of dopaminergic neuron death. Okay. All right. So let's move ahead. And I told you in Alzheimer's, you mostly see this as a cognition problem and a memory problem. In Parkinson's, you begin with the motor problem, but over the period of time, you also have non-motor symptoms, if you remember in the discussion that we have had. So that's the question here, and that should help you also understand the problems. So what is the first problem or primary symptom of Parkinson's disease? I think this is very easy to understand. Second one is, which of this is a non-motor symptom of Parkinson's disease? Okay, one is B. So Parkinson's motor means movement. Anything which is not to do with movement is non-motor. Any idea for the second answer? You can take a guess. Okay, C. And that's the answer, B and C. Great job. Okay. So, like I said, in Parkinson's, the primary symptom is muscle tremors. Along with this, you have rigidity. So, your muscles get rigid. You also have something called bradykinesia, which is basically slowness of movement. So, you have tremor, but otherwise, you have a very slow movement. If it's not shaking, you have a very slow movement. So, because there is uh, lack of control or lack of proper relaying of information, you have a very slowed movement, which should have ideally happened in jiffy. It's very slow movement. That's bradykinesia. That's a primary symptom. And other than that, you so all these other memory loss, headaches, visual hallucinations come way later in time. The first one is the motor symptom. And the other non-motor symptoms that are associated with Parkinson's is depression and sleep disturbance. So the question here was about non-motor. Everything else here is motor. So muscle rigidity is something to do with muscle stiffness. So your muscle is not moving. Bradykinesia, slow movement. So movement. Tremors, we already know it's about movement. So all of these are motor. The only non-motor symptom here is this mood hallucinations and your disturbances, all of these. Good. Next. Okay, now we come to treatment. Now I told you, after you knowing what is Parkinson's disease, how is it getting caused, you now need to know treatment. So what is the treatment of Parkinson's disease that was discussed in the lectures? Any idea? Any, if you can recollect? That's great. Yes, it was L-DOPA. The way also you can remember it if you have difficulty in trying to relate. Um, your Parkinson's, the main neurotransmitter that is getting affected is dopamine. So the treatment is also for dopamine. So you have L-DOPA. Before I tell you what that does, let me put it as a question. So L-DOPA is basically levorotatory dopa, so levodopa. What does it do? What is that therapy trying to do? Any idea? What is that? So your dopamine is getting affected. So what are you trying to do as a doctor when you're giving L-DOPA? What is it that you are trying to do? Are you inhibiting dopamine receptors, or are you increasing the synthesis of dopamine in the brain, or are you blocking acetylcholine release, or are you preventing serotonin reuptake? P? Yeah? Okay. So that's correct. And I'm guessing you thought in the absolutely correct way. So the first one says inhibiting dopamine receptor. You don't want that. We want dopamine in the body for its regulatory activity. If dopamine is not getting synthesized or dopamine neurons are getting killed or dying off, you want dopamine in your body because those neurons are not transmitting dopamine. So there's not much dopamine in the body. So you give L-DOPA, you want dopamine. L-DOPA is a precursor, it gets converted to dopamine. Now you have given the dopamine, which is required, and it does its regular activities. So you don't inhibit dopamine receptor, you want dopamine activity. That's why you're increasing the synthesis. And acetylcholine and serotonin is not something that is directly associated with Parkinson's disease, right? So if you're talking about Parkinson's, we are talking about dopamine, right? Good. Okay, now let's shift our gear from the second disease to the next disease that was part of your this week's lecture. The first one was Alzheimer's, second one was Parkinson's, third one was ALS or amyotrophic lateral sclerosis. So let's again start by answering what is ALS? Any idea what does ALS mean or what does it do from the given option? So let me read out the options for you. So amyotrophic lateral sclerosis affects sensory neurons in the dorsal horn, motor neurons in the ventral horn of the spinal cord, interneurons of the brain, and glial cells in the PNS. If you have any answer ready, it's okay, or else we can just look into the answer. I will wait for 30 seconds and then we can discuss the answer. Okay. So the answer is motor neurons, and this is again a motor neurons problem of the spinal cord and the brain. So ALS, sclerosis. So the term sclerosis generally means that hardening of tissue. So we have spinal. So this is a problem mostly for the motor neurons in the spinal cord. Okay, let's move to more questions so that it becomes easier for us to understand it. Okay, so a common genetic factor associated with familial ALS is which mutation? So basically, uh, we don't really know which reasons or what impacts the ALS, but we know one definite reason, and that was discussed in your lecture note. So if you remember that, that is the answer to this question. Do you recollect what was said in the class about this? Okay. So someone says B, and the answer is exactly that. So if you remember SOD1 mutation, we discussed this in today's lesson also. So let me give you the, wait a minute. Yeah, SOD is basically superoxide dismutase. And if you remember what we discussed about superoxide dismutase is the original function of the enzyme dismutase, superoxide dismutase, is to convert superoxide into hydrogen peroxide and oxygen. Basically, removal of the superoxide. Superoxide is a reactive oxygen species, ROS. ROS or reactive oxygen species. These kind of radicals, free radicals, or these kind of radicals which are superoxides, which have high reactive oxygen in them, they are extremely toxic for the cell because they react with the cellular intercellular proteins and they cause problems. They basically disrupt the cellular structure. So you have ROS, and you don't want ROS in your cell. That's why you need these kind of enzymes which remove the ROSs, different kind of reactive oxygen, among which you have superoxides, and superoxide dismutase removes these ROS. Now, in your ALS or in your SOD1 mutation, this dismutase is problematic. This is mutated. This has lost its function. So as a result, your superoxides are not getting removed. They are doing exactly what they're supposed to do. They are ROS. They will kill the cell. They will create problem for the cell. And when it's neurons, they will kill the cells, they will kill the neurons will get degenerated. Now, what happens in this ALS is, because you have a muscle and your muscles have a lot of neurons, so as your neurons are dying, your muscles no more have the action or uh, crosstalk with neurons. So muscles are not getting used. The muscles are hardening up. They are getting atrophied. This is called sclerosis. So your muscles get degenerated over time. So ALS is when your muscles get impacted. Your muscles getting impacted means you can't do any motor activity. So your motor neurons are getting affected. Your motor pathways or your motor activities are getting impacted in your ALS. Right? Okay. Okay. Now we have spoken about neurotransmitters in all of these, right? Now, which neurotransmitter is responsible for ALS pathophysiology? So we know about acetylcholine, we know about dopamine. Which one is responsible for ALS? So the word here is, so the word also tells you the mechanism by which it plays a role. So excitotoxicity. So the motor neurons get excited, more excited, excessive excited, basically excessive action potential, and they get damaged. They get damaged over time. So which one of them get excited? That's how you remember, and exactly how one mentioned. It's glutamate. We know glutamate are excitatory neurotransmitter. So more the glutamate, more the excitation. Very good logic, right? Okay, now coming to the symptoms, so we know how it is done. Let's look at to the symptoms of ALS. So which of these is not a symptom of ALS? The other way around. So you have progressive muscle weakness, you have loss of voluntary motor control, you have significant sensory deficits, and you have muscle atrophy. Which one is not for ALS? I told you about the general flow of problems. So which one do you think is not associated with ALS? So I told you about it's connected to motor neurons. I told you the motor neurons are connected to muscles. Motor neurons are damaged, so the muscles are also damaged. So you have already got the uh, sorry. I want to know about ALS. Are you saying that you want to discuss more about ALS? Okay, we'll do that. Uh, Let me finish all the questions about ALS, and at the end of that part, I will summarize these questions for you. Okay, so just bear with me till I complete ALS, so that I don't want to give any information before you see the next question, so that it doesn't get repetitive. Just bear with me, and I will explain to you ALS right when we end all the questions for ALS, which is probably in a slide or two slides. Okay. Okay, so here we have significant sensory deficits. So in ALS, your motor neurons are getting affected, right? So everything that is to do with movement is getting affected. And everything that is to do with muscle is getting affected. So muscle is getting weak, your muscle is getting atrophied, basically it's losing its shape and size, and you have motor controls problem. But not for sensory. The sensory neurons don't get affected in ALS. Okay? Okay, the next one is, now I told you every disease, we are looking from the perspective of what is the disease, how is it causing, and what is the treatment. So the treatment here is the next part that we are discussing. So the first question is this: riluzole is associated with which disease? Obviously, because it's part of the question here, it is for Parkinson's. Oh, sorry, ALS disease. I will now. Yes, correct. Now let's do come into this. So, which of the following is an improved treatment that helps manage the symptoms of ALS by reducing glutamate-induced excitotoxicity? So, as you can see, we spoke about excitotoxicity. So, your treatment basically targets those very problem. We talked about glutamate being responsible for excitotoxicity in ALS. Now we are talking about a treatment for targeting glutamate excitotoxicity. Yeah. Any idea which would be the correct answer here? Okay, no worries. The answer would be riluzole, the one that I just discussed. So what riluzole does is, so your it's riluzole. Okay, so what your riluzole does is it targets the glutamate excitotoxicity, right? More the glutamate, more your motor neurons are getting excited and they're getting impacted. Okay, so what these riluzole does is it basically stops the secretion of glutamate, and if your glutamate is not released, your glutamate cannot like activate the or generate the action potential in the muscle. Okay. Okay. Now, since you want me to discuss ALS once again, just give me 2 minutes. I'll just go back to the slide of ALS and discuss. Okay. Yes. So I will come back to SOD after this. Okay. So this is ALS. ALS, as I told, it's about your amyotrophic lateral sclerosis, right? So what does says is that there is a sclerosis, which is hardening of your muscles, and that's because your motor neurons are getting affected. Okay, this is the progressive neurodegenerative disorder, like all the other disorder that we have been discussing. And what all gets affected? It preliminarily first affects the motor neurons, right? And the motor neurons, as we know, are present in both brain and spinal cord. So both the brain and the spinal cord motor neurons get affected. If you remember, in the brains, it's the upper motor neurons, and in the spinal, it is the lower motor neurons. But what you need to remember is both brain and spinal cord motor neurons are getting affected. Okay. Now, these motor neurons, which are present in your, so you can see in this. Okay, so these are present in different regions. They are present in your motor cortex, brain stem, spinal cord. Over the period of time, they die. Okay, we'll see why and we'll see that later, but it dies. So now see this. This is a normal cell and normal nerve cells, neurons, and you have a muscle. So you have innervation. What does innervation mean? You have an impulse here, muscles get activated, and your muscles contract. Remember all that action we discussed, right? You get an impulse, you start moving your hand, or you get a knee jerk reaction or something. Your muscles is impacting based on the neural input. Now, in case of ALS, your this neuron starts dying off, so it dies off. Now, what happens? This muscle doesn't have any input, so it is not getting used at all. So a muscle, if it is not used, it basically starts losing its function. It's a wasted muscle. So it also starts degenerating. It gets weakened and basically starts getting atrophied. It basically starts losing its shape. And if you see this muscle and if you see this muscle, you look at the difference. It has lost all its cells. It has started to weaken. So your muscles are also getting impacted. So from your motor neurons, your muscles are also getting impacted. So in your ALS, both motor neurons and muscles are getting infected. And so what? So overall, if you see, so what happens is that you have a scarring of your spinal cord pathways. Your muscles are not functioning. So your spinal cord and your brains are not able. So there is no proper relay of information and you have hardening of muscles there, right? And what are the symptoms? Because your muscles are not functioning, you will have cramps, muscle cramps, you will have muscle weakness, and then because your motor neurons or neurons are dying, you will have, over the period of time, you will have difficulty in speaking, swallowing, all the problems that we have spoken, not of memory though, but everything that is involving some kind of muscular movement. You're swallowing, you're breathing, you're speaking, you're using muscles, and because your muscles are getting affected, you will have all of these difficulties, right? Right? I hope that makes sense for ALS in general. And now, see, that's why, because your neurons are dying, what exactly is causing the neurons to die is not very clear. There are probably many reasons there, but we don't know. This was what professor was trying to tell you. But we know one reason that is directly affecting ALS, and that is your SOD, which is superoxide dismutase, where you have the superoxides in the cells, which is causing oxidative stress. Oxidative stress, like I said, it hampers cell in different ways. There is protein misfolding, there is glutamate excitotoxicity. And because we know this reason, for your ALS, for example, let's say SOD mutation, or we know, for example, glutamate excitotoxicity, we can target these two and try to reverse the actions. But there are many other cases where we don't know what are the reasons for your ALS. So that's why it's not really treatable unless that person has SOD mutation that you can treat, right? So that's complicated disorder about ALS. One of the reasons is superoxide dismutase. This is not the only reason. I hope that was clear. If you have any other doubt, please put in the chat box, and I will just quickly go to the next question so that we move ahead with the class. Or if you have any doubt, please put in the chat box. I am always ready to answer them. Okay, now we have completed ALS, like I told, and I will now go to the last disorder or disease that was discussed in the lecture, which was spinal cord injury. So, what basically is spinal cord injury? We discussed today in the class that spinal cord injury is also very important, and we need to understand why. So let's begin with these questions, and let's try to understand what do you make sense of spinal cord injury. Okay, let me give you the answer. So the primary pathology in neurons due to spinal cord injury is axonal degeneration. Let me tell you what happened in spinal cord injury. So in spinal cord injury, we are referring to anything that is affecting your spinal cords, right? And if you can imagine what would affect spinal cords, any kind of accident, trauma, accident, or any kind of violent activities or falling down, which affects spinal cords, right? If not trauma, you can have tumors, you can have infections in the spinal cord. Now, spinal cord is innervated with many neurons. When the neurons also get affected, that's when your problem starts showing up, because spinal cord, if because we are particularly talking about neural systems here, so the neurons of spinal cord getting damaged is when we term it as spinal cord injury. And there are a lot of problems associated with these neuron damage. We'll see how. Okay. So we just saw that is axonal degeneration. We'll define it a bit more, right? So in your spinal cord injury, which of these now will be primary pathological event? So if your spinal cord is affected and your motor neurons get, or your neurons get damaged, what will happen in the spinal cord? Or what will happen as a pathological event? Any faint idea? A. Well, it's B. So you don't demyelinate neurons. There is no demyelination. Demyelination is basically removal of myelin sheath, right? That's not what's happening. That neurons is damaged. So what is happening when neuron is damaged? You have these immune cells which come to play. They come to rescue the site of injury, and you have inflammation, which is a normal response of your body to any injury that you have. So in your spinal cord injury also, you have the influx of immune cells. That's the beginning of inflammation when you have a spinal cord injury, right? And when these start infiltrating, there is something called blood-brain barrier, which we had discussed. We can see in the recorded lecture. So your blood-brain barrier is also disrupted in spinal cord trauma, and you have inflammation, which gives rise to secondary problem, which was also discussed earlier. But we have questions, so let's see what happens there. So I told you about primary damage, that is your motor neuron damage or neurons damage, and neuron damage by having inflammation. Now, once your inflammation has happened, or which of these now will be your secondary damage? I just discussed that. So it should be giving you away the answer here. Any idea? So basically, if your neurons are damaged, you have these immune cells coming into the site. So if your immune cells come, what is the first clinical, like outcome of it? Symptoms of that process. Okay, you say acetylcholine synthesis. It's incorrect because it's a secondary damage, and when you have all this, you can imagine this from your regular injury also. If you have an injury, let's say a cut is there, you will have inflammation here. Why? Because your immune cells will come and start to accumulate here. So the accumulation of cell will lead to inflammation. That's exactly what is happening here. So you have inflammation, and you have blood brain barrier breakdown, which is a problem of spinal cord injury, because your spinal cord, the main, see, so the lot of neurons are here, right? If it is getting injured, so many pathways are getting impaired. Okay. Okay. There was a term that was used in the lecture, which is called syrinx, right? So what is this syrinx when we are talking about spinal cord injury? Any idea? Anything that you can recollect? Yes. It is a fluid filled cavity with spinal cord, within the spinal cord. So your uh, basically all this inflammation and everything, along with this, there will be a fluid cavity with this, with cyst. Okay, so it's a cavity which is trying to protect your fluid, which is trying to protect the spinal cord from further damage. So you have this syringe formation also, and with a lot of cells which are taking part in damage control. Okay. Okay, now, which cell types plays a major role in forming the glial scar after spinal cord injury, inhibiting axonal regeneration? So in your spinal cord, axons are getting damaged, right? Glial cells are also immune cells. So when you have a glial scar, which is basically problem with your glial cells, it means your neurons are not able to come back. They are not able to regenerate. So the question is, which cell is responsible for this scar formation in your spinal cord injury, which is basically when you have the glial scar stage, which is at the very end, then you cannot have axon regeneration. And the person, because spinal cord injury is seen as paralysis, so you will have paralysis in that case. Are Did you mean? Okay, let me give you the answer. Oh, you're typing again? Yeah, B. Astrocytes, right? So you will see when I will share the lectures with notes with video with you. We have discussed about all these different kind of glial cells, astrocytes, oligodendrocytes, Schwann cells, and among that we have seen that astrocyte plays a role in immune cells, and this is a cell which comes into the play of immune. So that's why these are the secondary damage, because these are the cells which are coming to the site of injury, and then they are causing the scar because of glial cells, and because of that your neurons are not able to regenerate, and because the neurons are not regenerating, you have the spinal cord activity is not coming back into action, and this is generally seen as paralysis in most of the patients. So you will often hear them, they are paralyzed from the lower end or upper body or left side or right side or the whole body, depending on what is the extent of damage to spinal cord. Okay. Yes. Now again, similar to all other uh, injuries that we have so far or diseases that we have read, which neurotransmitter is part of pain signaling spinal cord injury? We know acetylcholine, we know dopamine. There are two other options. Which one do you think is part of your spinal cord injury, if you can remember? And remember, we are talking about pain. So which one would be more suitable there? Think. This is very easy to, if you keep a logic, pain means something is getting activated. That's why it's some is getting activated. Which thing will have a direct impact on activation or relay of transmission of signals in the neurons? Any idea? Let me tell you. So it's glutamate, right? So glutamate, because I'm talking about pain signaling here, if you have excessive glutamate, you have excessive excitotoxicity. You have excessive excitation of your neurons. They're almost persistently activated. So your pain is in chronic pain stage. And uh, so what you are doing is you have these NMDA receptor. If you remember, glutamate responds to NMDA receptors. So your receptors are getting over activated because there is over production of glutamate. So all the receptors are activated together and you have extreme pain in spinal cord injury. So that's how you see person who has spinal cord problem will have extreme pain. That pain comes from the activation of the receptors, and that's because you have excessive glutamate production during spinal cord injury. So you see everything is linked. If you just start joining the link, everything that you see in movies or real life starts making sense, right? Okay, the last question on injuries here. So which of this is not a standard treatment? Just like we have been doing for all others, we'll finish it with treatments. So which of this is not a treatment for spinal cord injury? And then whatever is the treatment of spinal cord injury, what is the primary therapeutic goal? What do you want to achieve with this? So imagine your spinal cord injury to be extremely high pain inducing, right? So what you are targeting here is, the first one is saying which is not a standard treatment. So you have corticosteroids, which reduce inflammation, surgical decompression, and physical rehabilitation. So these two are for immediate effects. Your spinal cord injury has to be reduced. So first you target the inflammation, then you target the decompression. Decompression is basically getting it back into shape. Inflammation is basically when they have swelled up, right? So you're trying to get them into shape. And once that is done, you will have a lot of physiotherapy session. You have a lot of motion related treatment for spinal cord injury patient, which is physical rehabilitation, right? So this is what people generally use for treatment of spinal cord injury. Gene therapy, which is to remove the damaged DNA, is something not yet into the action, right? But all of these three basically wants to just reduce the inflammation, because everything that we have been talking about, inflammation, pain, is because of the influx of cells, and the treatment wants to target those very effects, right? Okay, so we are done for the day. I will just quickly go over the question that I couldn't discuss in today's session, but I will wait in each of these slides so that you, when you are practicing or you're revising, you get to see these questions. So I have all of these assertion reason based questions. What I would like you to do is you would read the question, pause and think the answer, and then check for the answers. Most of these questions are self-explanatory, so they don't require explanation. But if one of these questions require explanation, they are in the slide. I have given them as notes. Okay. So yeah, these questions I had don't, due to paucity of time, I have not been able to discuss, but I am showing you in the slide so that it's in the same video where you can directly look into the answers, right? Okay, and that brings us to an end of today's class. Next class, we will do more about neuromuscular junction and then quickly move on to, I think we have a hearing system. So interesting systems, and switching from the normal neuromuscular junctions and skeletal muscles discussions, right? Okay, so we stop here today and we meet each other in the next session. Thank you so much.